set my phone to silent. Accidentally happened.
I need it for the timer.
Good morning, everyone. May you please kindly take your seat because we're going to start soon. All right. Well, welcome everyone to this very unique and interesting event. Let me briefly introduce myself. I am Barbara Yu, the co-founder and president of the Citrin Foundation. We are a non-profit organization focused on finding a cure for citrin deficiency, a monogenic metabolic disease. And I am very honored and pleased to host and sponsor this event today. Here with me is Cary O. We are co-chairing today's event. Cary is from Oregon Health and Science University and who has a ton of experience on many of the issues that we will touch upon today. So why are we hosting this roundtable? What is the purpose of this discussion? As we all know, there has been fantastic scientific advancement for novel therapies available for monogenic metabolic diseases, ranging from enzyme replacement therapy, mRNA therapy, gene therapies of all kinds, and novel small molecules. However, when we look at what is happening now, very often, these therapies get to patients either too late, takes too long, or is extremely costly. And worse still, many of them don't make it to patients. And we must wonder why that is the case. Many of you who are involved in this daunting process of developing novel therapies to patients will notice that there are many challenges and hurdles along the way. It's a complicated process with many stakeholders involved. And therefore, we decided to bring together a group of very relevant stakeholders to discuss what are these challenges, what could be the potential solutions, and if there's no solutions yet, maybe how we can forge a pathway forward. Just to give you the context of some numbers, the cost of developing rare disease therapies is roughly half of that of a common disease, probability adjusted. However, the addressable market of rare disease is several thousand times smaller than common disease. If we don't do something to make this process faster, cheaper, investments into this area will slow down or stop. Or there will be therapies available that's extremely costly to payers, to governments, to families, to healthcare system. Ultimately, it is patients who will suffer. And patients is at the heart of everything that we do. So, without undue delay, let me introduce today's panels. First, joining us, is it joining us? I will introduce the people here first. Oh, okay. First joining us are the top two global regulators who regulate many of these matters. And on the screen is Ms. Kristina Larsson, who is the head of Orphan medicine at the European Medicine Agency, EMA, based in the Netherlands. Soon, Dr. Peter Marks from the FDA, and many of you who know, who is the director of the Center of Biologics Evaluations and Research, will be joining us as well. And from here, we have clinician scientists who has been doing multiple clinical trials, working very hard on developing these novel therapy in a preclinical setting. First, we have Dwight Koeberl, who is from the Duke University School of Medicine. And then we have Simon Jones, who is from the University of Manchester. But very importantly, we must remember all these therapies are for patients. So we invited 2 patient advocacy groups heads to join us today. First is Teresa Warner, who is the president of the National Urea Cycle Disorders Foundation based in the United States. And then there's Raquel Marquez, who is the founder and head of the San Filippo Portugal Association based in Portugal. What's more unique about them? And like me, they are parents of a child with metabolic disease. These issues are close to their heart. Now, let me give you a rundown of today's event. First, Cary is going to give a brief presentation on what are the phases of the drug development process and the possible choke points. And then we'll open to the discussion divided into 5 topics. First topic is how do we translate preclinical findings to successful clinical trials? Second topic, how can we effectively use biomarkers as surrogate endpoints in clinical trials? Third topic, how do we design successful clinical trials? Fourth topic, how do we increase patients' participation in these trials? And finally, we'll look at what is the advancements in the regulatory framework and how can we work together globally as one community. So thank you very much, and without undue delay, I'm going to hand over to Cary.
Thanks so much, Barbara, and thank you for inviting me to participate in this. I think it's very important. If I could have my slide deck, please, on. Just because I have some conflicts, I do have participated in several clinical trials for various different organizations and some preclinical work, and none of the companies really have contributed to any of my remarks that I want to talk about today. So it's maybe hard to see from the back, but I just recapitulated sort of the standard preclinical and clinical trial design as it currently stands from preclinical up through phase 4, and everybody in the room knows this, so I'm not going to go through it in detail, but mostly it concentrates on adults. That's been sort of the standard paradigm, and the only wrinkle being for rare disease that sometimes instead of doing phase 1 in healthy adult volunteers, sometimes we combine phase 1 and phase 2 in adults with the disease, if it's a biologic that doesn't make sense to do in healthy adults. But this is challenging, as everyone here knows, in the rare disease space for a variety of reasons. So often we have trouble finding sponsorship and raising funds for preclinical development of a clinical candidate drug in a rare disease space, often because there doesn't seem to be a good, clear reason for profit at the end of this. Many diseases have effects predominantly in children, and there are very few affected adults, so the standard clinical trial design doesn't really fit. There are too few affected individuals to support the traditional trial design. That's a major issue for a lot of our disorders. There are often ethical concerns, and a lot of the families share this a lot, ethical concerns of doing a placebo-controlled trial, particularly in children, and particularly if there's only a few individuals with this disorder, and particularly developing outcomes for trials. If it's a multi-year progressive disease, it's very difficult to do a traditional clinical trial design if you have to wait many years and follow the progress of outcomes. And so these are just a few. There's other challenges, but these are sort of some of the major disease challenges that we have for fitting our rare diseases into this clinical trial framework. So just to set that as the stage and some of the concepts we wanted to cover, we'll get straight into some of the discussions. So we can go back to having the Zoom participant on, please. So our first topic was topic 1 is to translating preclinical findings into clinical trials. Our key question really is how do we bridge the gap between preclinical studies and clinical trials in rare diseases, or as we've termed it, overcoming the valley of death and getting our preclinical work into the clinical space. We've asked Dwight Koeberl and Kristina Larsson to comment on this. And so Dwight, do you want to start off?
Sure. Yeah, so I started a clinical trial of gene therapy in Pompe disease in 2019, so I'll speak from that experience. So there are a few challenges, and probably number one is that academicians like myself just don't have regulatory expertise. So it's kind of a do-it-yourself program, and it took me 10 years really from the concept to filing the IND. Then secondly, sponsors, they really don't understand rare disease. So there's an education process, there's a lot of collaboration that's needed. But along the way, there were sort of three key limiting steps. One was the farm tox study to present to the FDA, and that was supported by a federal program called GTRP. Would have been very expensive, we couldn't have done it without that. The second was actually filing the IND, and it wasn't funded, but it was supported by discussions with a program called TREND, again at NIH. And they basically encouraged me to file the IND, even before we had the manufacturing, which I didn't realize was possible. The third thing was finding a sponsor for manufacturing, and that was an industry sponsor. But for various reasons, we couldn't find that sponsor in academia, and we didn't have funding for it, so industry was really vital. And everything else seemed to follow from that. Funding finally came through on the 6th try with the grant application. But so that's my experience and how I navigated it.
Kristina, do you have any comments on this process?
Yes, thank you. Maybe I should first ask, can you hear me?
Yes.
Okay, excellent. It is a bit funny to be here online, I have to say, but I'm very pleased to be in the session. And yes, of course. Now, when it comes to preclinical studies, the regulators are mainly interested in safety. Of course, we're interested in the toxicology and any problems that, there's a whole package, and I'm sure that Dwight has now learned a bit more about that. Everything you need to do before you can actually file a marketing authorization, but mainly it relates to safety. Now, so I work with orphan medicinal products at the European Medicines Agency, and when we have designations, so this is a program that can be done very early, we do like to see some sort of not toxicology endpoints in the clinical studies, but rather maybe if there is possibility to see something of an endpoint that is similar to what do we expect in the patients later on. So if it's survival or if it's reduction in a particular biomarker or whatever. So we would like to see that. But how is that then translated into the patients? That is obviously very difficult. And I don't think that we actually have an answer to that, how to actually anticipate what you see in animal studies and what then would be the result in patients. I wish we had, and I think we all wish, that if you see a survival in a mouse, that you would see that in patients as well, but it's not always the case. Now, I was quite interested in Dwight's mention of that it may be difficult, as specifically if you're at university, you may not actually know the regulatory procedures, etc. At the European Medicines Agency and at the national competent authorities also in Europe, and I know in the FDA as well, there is the possibility to discuss with the regulators rather early. So here we now have a new academic office. So if you are an academic group or a single academic person, you can actually contact the agency and get initial guidance, just try and understand a little bit what is required. And it's probably good to do at a very early stage. But I think what is important, and maybe that's something that you can also try and disseminate between your colleagues, etc., is people don't even know that it is important that what you do with your non-clinical studies, that you can then use that later on in the regulatory process. because would be a bit sad if Dwight did a lot of non-clinical studies and then, the FDA comes and says, oh, good that you did it, but we're not interested in those results. We're not interested in what you have done there. So to know upfront is obviously quite useful, because then you can plan even your non-clinical studies and ensure that the funding you manage to get is actually spent wisely. And with regards to funding as well, yes, I think it's very difficult, and I'm obviously not in a funding position at all. Pharmaceutical industry find it, I think, challenging right now, but there are possibilities also in Europe from the European Commission. They have specific funding programs for rare diseases, and not only clinical trials, but actually rather early development as well. Should I stop there, maybe?
A quick follow-up.
Go ahead.
Yeah. So as Kristina mentioned, actually, that regulatory support is really key in the study design, in being sure that you'll address the concerns of the regulatory agencies. So I was very fortunate to have a regulatory expert at Duke, Bruce Burnett, who really helped me with the study design for the pharm tox, and with contacting the FDA for the pre-IND meeting, where they reviewed our study design, they endorsed it, and then we had a good plan for going ahead. That's really key input to have from the start.
Simon?
Thanks. Just from, I think these are really important points. To be maybe slightly controversial, one of the challenges in being an academic and developing programs like Dwight, we've done similar programs. They take a very long time. I think we're both experiencing that. And we tend to, as academic groups developing our own therapy, deal with the regulator that's in our sort of backyard, as it were. I'm from the UK, Dwight's from the US, but developing rare disease therapies is a global enterprise. Our patients come from everywhere. The future marketable population, to use company speak, has to be a global one. And so it's pretty challenging. It's hard enough to speak to your own regulator when you're an academic, never mind all of the regulators in all of the countries. So that's a kind of challenge and a real risk for us. And the other point is, yes, these preclinical toxicology studies clearly are important. We're clinicians as well as academics. We don't want to do harm to our patients. But many of the therapeutic modalities, I work mostly in lentiviruses. I think you're mostly in AAVs. And, there are lots of other lenti products. There are lots of other AAV products. But as small academic groups, or even when we work with small biotechs, they don't have access to everybody else's data. And so there's a finite pot of money in rare diseases, and there's a lot of duplication, because if I'm submitting a lentiviral program, I have to do preclinical toxicology. That's not work you can get funded at an academic grant, because you can't publish that, and it's not a cutting-edge paper. And yet someone else has probably done that study. And so it feels like in this finite pot of money that we have to develop therapies for these multiple hundreds, thousands of diseases, we're just duplicating a lot of this effort. And I don't know how we get past some of that issues around confidentiality and ownership and IP, but it feels to me that's a challenge and an obstacle.
Just one more thought after that. So through this GTRP NIH program, I was actually able to cross-reference toxicology packages, other toxicology packages. So we only had to do a mouse study, which was really pretty unique. But given all the experience with the vector system, we were able to, reference that data. And sharing data is really key to moving the field ahead. I agree.
So you did have access to even commercial toxicology?
It was actually CHOP's data, so an academic lab, and because they were participating in GTRP as the core lab, they shared the data. It was really invaluable.
But it was purely academic data that had been already generated, you think?
Yeah, it was.
Okay.
Good point.
We don't, and best I know, we don't have an analogous global, even academic repository for that type of data. We don't have effort like that, right, that we know of?
So there's an initiative, again, coming out of the US with Critical Path Institute, who work with, I think, the EMA as well as the FDA, and they're trying to be a sort of neutral repository of lots of data, initially with biomarkers, also hopefully with natural history data, because again, there's huge and probably unethical duplication in natural history studies in some of these exquisitely rare diseases, and they are trying to be a repository for data where academic groups and industry could put their data for more widespread use, but it's very early days, and I don't know whether that will be successful.
Yeah, and I know,
Yeah, it is indeed early days, and we have been working on that as well, and they presented quite a lot. So there is in the CPath repository both data from European sites, but also from the US, of course. But yes, you then as an academic group and companies will have to be willing to share that information. That's
and I know, and
relies on that.
Peter's going to be joining us. We should probably ask him
Yeah.
about this. I mean, they do have this new platform initiative, right? Whether that would be, I don't know if that really had included this sort of potential for being able to build on others, you know, safety data, but that would seem like a logical thing to use that for, right?
Yeah. So it's mostly natural history so far, and it's been incredibly difficult to get people to share. That's exactly the right point. And academic groups have been at least as unwilling to share as industry. I have to hold my hand up there, and I think the biggest changes that I've seen driving that have been from the patient advisory groups, patient advocacy groups, driving that, because actually they're the only, while they're always viewed as biased and subjective, are actually by far the only neutral partners in that initiative from my perspective.
Yeah. Should we move on?
Okay. Yeah, so the next topic is the role of biomarkers. And as we know that there are many beautiful biomarkers we discover in labs. But very often when we translate to clinical trials, there are difficulties of using that as clinical endpoints. So we would like to open this topic to Kristina first. And Kristina, if you could comment on this, thank you.
Yeah, and I think you know that, of course, as the regulators, we do accept biomarkers or surrogate endpoints. We do it already, but maybe not in rare diseases so much. You already were in diabetes, we do it. In cancer, we do it. Why? Because in those situations, there has been at least, I'm not going to say undoubtedly, a link between showing something in a biomarker or a surrogate endpoint and the actual clinical benefit for the patient and the outcome, which of course, thinking of small patient population, it's more difficult in small patient populations to do. And I guess that's why we haven't really seen it so much in rare diseases. I think because since this group is very much into lysosomal storage disorder and monogenic diseases as well, there we do have enzyme replacement therapy. One would assume that, for example, if you can use an enzyme replacement therapy, bring down the biomarker that you're interested in, that it should have an effect in patients. And also maybe if you even have a good animal model, that if you show that, it should also translate into patients. And we do use it as secondary endpoints, but I had a look back, we haven't really used it as a primary endpoint in any way or form. And I was in another meeting also with, not in this particularly, was on inherited retinal dystrophies, but they had the same ideas. You know, we have biomarkers, we don't want to wait too long to actually see the results in patients because we would have to wait many years to see it. It's difficult. But on the other hand, you may also don't want to expose then patients to something that you don't necessarily know if it works. So I guess this is where maybe as the regulators we are reluctant, conservative to say that we would like to see something that is actually beneficial for the patients. And since there are patients now in the panel as well, we may want to ask them with regards to sort of surrogate endpoint. But one thing that we do have as a possibility, both at the European Medicines Agency and at the FDA, and I'm sure other agencies as well, we have sort of a qualification procedure. So you can come to the agency and get a surrogate endpoint or biomarker qualified, but you would have to validate it then and show that actually, if I can show this type of reduction in this biomarker, it will actually have this type of impact on the patient ultimately. So it's a challenge. We have done a few of those, but yeah, it is a challenge, I think specifically for rare diseases because of the limited patients. It would, of course, be even more beneficial for small clinical trials, which we'll come to later, if it was possible to show something early. But generally, we are open to it if it is possible to link the biomarker with some benefit for the patients.
First, Simon.
Well, no, I don't know if you guys want to respond directly to that.
When it comes to benefit to the patients, of course, when you're recruiting for a trial, knowing what those benefits are up front is how you get patients into the trial. But also being able to adjust is also important. So if there's a way to do that and to make an adjustment, I think patients would value that.
Can you explain more? I'm not sure quite I understand what you're...
Well, understanding the endpoints that are valuable to patients when putting together the trial. Maybe the endpoints that are presented are not what patients are most interested in.
Right. So my add to that is that, you know, we are a patient organization focusing and funding research for finding cures. One of the challenges we have is that there's not many research done. So then we fund the research, and then you demonstrate that this biomarker will correlate to the disease progression in a biochemistry or lab basis. Then we roll it out to patient, but it will take years to validate it, to link this particular biomarker to this particular clinical endpoint, and then to do the trial. So if we go on this waiting process, the trial wouldn't even happen in 10 years' time. So that's why we wonder, is there a way for us to say, okay, this seems to make sense scientifically, biochemically, it makes sense. And the patients also are educated on it and say that, okay, explained and they understood this biomarker possibly correlated to this clinical endpoint that you want to see. Do you accept this as an endpoint in the clinical trial? If the patients say, yes, I will accept it because I don't want to wait 5 years for this biomarker to deteriorate and therefore my condition deteriorate. And at that point, can the regulators accept it? Is that it's kind of similar to what you're saying, yeah.
Yeah, so, and indeed we may, but there are procedures to discuss with the agencies up front if you have a new endpoint that hasn't been used before, for example, or maybe it's a rather standard endpoint that is always captured anyway. Both the European Medicines Agency and the FDA, we have some procedures at the European Medicines Agency, we have something called a conditional marketing approval, where you can have more limited data at the time of marketing authorization. So it can indeed be based on a surrogate endpoint, and then you will have the conditional marketing approval, collect additional data, maybe continue the same study to get that type of endpoints that we're looking for, the more clinically relevant endpoints. I don't know if you've decided that the patients and the clinicians think this is really important for the patient. You decide at the time of the conditional marketing approval how long this will take. Every year you review, maybe in 3 years you say, we want to have that final endpoint. And then if then it is shown actually that you've reached the clinical endpoints that you were looking for, then this conditional marketing approval can be changed to a normal marketing approval instead. So that there is definitely a possibility to do this. But again, the first biomarker in that case, or surrogate endpoint, has to be something that is generally accepted, I think, by the community, the research community, the treating physicians, that this is actually something that is related to the disease.
I can add, for example, for neurodegenerative diseases like Sanfilippo syndrome, as the general MPS neuropathic disorders, these diseases are slowly progressive diseases. So it takes a lot. It takes several years, usually 8 to 10 years, to show to the progression takes so very long, and it is very difficult to track the changes in such a long period of time. So clinical endpoints are very, very challenging in these diseases. So I can say that, for example, for Sanfilippo diseases, a therapy has been in the works for 20 years now, and 10 companies have dropped their programs or shelved them because they don't meet the needs of the regulators to show the clinical endpoints, because the disease is very, the symptoms are, there is a great variability of symptoms, and the patient population differs a lot. So, it is tricky. In Sanfilippo disease, heparan sulfate, for example, is the underlying cause of the disease, and it is used in diagnosis. It is what is used to control the evolution of the disease, and is what monitors somatic treatment response. So it is not the gene or the enzyme that causes the disease, it's heparan sulfate. And we have now a way, the science has evolved to measure CSF heparan sulfate in the brain. So I think if we don't choose more innovative pathways on the regulatory process, we were still being on the same stage that we were 20 years ago. We were full of expectations with a promising pipeline, I can say 5 years ago, and nowadays we still have nothing. And if nothing changes, and if the regulatory don't find another more flexible approaches or innovative approaches, the rare diseases are lost.
Thank you. Yeah, so in terms of the role of biomarkers, you know, primarily before I'm a researcher, I'm a pediatrician. Before you guys are patient advocates, your parents, and so none of us value biomarkers more than people. We only move to biomarkers if we have to. We all want to see meaningful clinical endpoints. But in the neuronopathic lysosomal disorders, we've seen over the last 10 years, probably more than half of the novel therapeutic programs disappear. And by my calculation, it's not published, but you know, I think only about 10% of those failed programs clearly didn't work. And the rest folded and have disappeared, perhaps forever, because it was just too hard. And that's a disaster for our community. And if it carries on with that rate of failure, then it's going to be incredibly difficult to entice biotechs or even sort of medium-sized pharma companies to come in and help us move, our small academic programs in 1 or 2 centers and make them multi-center trials and then, actually license drugs, which is what we want. We need to get drugs to patients. And so in the neuronopathic MPS disorders, for example, as Raquel was saying, I've worked in Sanfilippo trials for 18 years now, and we've seen multiple programs fail. When I think about the perfect trial, it's getting patients identified at birth, following them up for maybe 10 years with an equally matched control group who don't get any treatment, and we watch them decline towards a sort of vegetative state after 10 or 15 years, and then we might have perfect data. And I don't think anyone thinks that that's an acceptable, ethical, or financially viable. It doesn't work on any level. And so, how do we manage this? And I don't think there's any way to bring forward a new therapy in that program, in that area, in the neuronopathic MPSs, without a conditional or accelerated approval that is based on a biomarker that I think we all acknowledge is likely to be imperfect, but the best we have. And I think a conditional approval or accelerated approval is likely to have risk associated with it. And I think we all accept that risk. We all want there to be a clinical endpoint. I don't want to treat a patient for their CSF heparan sulfate. I want to treat a patient for greater benefit. But it will take me 10 years for that individual to know whether there's actually real meaningful benefit. I would, to be able to validate some of these endpoints with multiple regulatory agencies would take data that I don't think I could develop without actually having a therapy. So we've never had a successful therapy for Sanfilippo or the neuronopathic MPSs. So we don't know the true impact of reducing heparan sulfate from very early on in life and then following that patient for 10 years. We can't know that until we have an effective therapy, but we can't develop an effective therapy without us being able to use a biomarker that uses that. And so there's a real chicken and an egg problem, I think. And at least for the neuronopathic MPSs, we have a common biomarker that we haven't even validated yet, that we can share for multiple diseases. But there are so many monogenic disorders where the biomarker will be specific to that disease. And how on earth are we going to manage that? And I think the validation route is maybe possible for much more common diseases, but really worries me that academic groups and small biotechs and patient organizations could spend many years failing to achieve a fully validated biomarker and not bringing forward therapies. And I'm not sure how that serves us. And maybe the language that is used more recently by the FDA, talking about reasonably likely, that a biomarker needs to be reasonably likely to be associated with clinical outcomes, is the best in our community that we can achieve. Bearing in mind, of course, that we have monogenic disorders, so the role of the biomarker is likely to be much more biologically plausible than in common diseases where the etiology is much more complex and diverse.
That's a very good point. I would like to welcome Dr. Peter Maas from the FDA joining us from the United States. It's very early for you. I really appreciate that. Then I will hand over to Dwight. I know you have some comments to make.
Yeah, just a couple. But so in metabolic disorders, we're fortunate to often have a biomarker that's related to the enzyme deficiency. And we usually have validation in the animal model. And, that's the biomarkers improving as, say, muscle function improves. So it would be really important to understand what's needed for clinical validation of the biomarker. Would it be in a successful phase 1/2 study, you see improvement in the biomarker as a secondary endpoint, and then it could be used as the surrogate endpoint in the definitive study, perhaps? I don't know if we'll answer that today. Another thing I just want to mention, a concept that's been put forward by Crystal McCall and Don Cohn in California based on the experience of gene therapies being pulled off the market because they weren't profitable. So they've suggested a not-for-profit entity being formed that would raise funds from various sources and be dedicated to gene therapy for rare diseases, do the late-stage development and marketing. And if that were successful, it would be a way for these gene therapies to continue without the pressure of being profitable. So I wish them success in that. That would be a really beneficial thing to do.
Thank you. You know, we should move on to the next big topic where we will open to the floor, and it's very exciting. So, Cary, you want to lead that?
Yeah, so our next topic was still relates to that, but it's designing successful clinical trials. So what are the specific challenges and solutions in designing successful clinical trials for rare diseases? And we've actually asked Peter to lead that conversation. So Dr. Marks?
Yeah, no, thanks very much. So, you know, for us, you know, a lot goes into the planning of successful clinical trials. You've already talked about some of the issues that, even though I joined late, I could hear this, that I'm figuring out what is the fit for purpose trial for a given disease is part of the issue. There's been a lot of back and forth about whether we at FDA need to have randomized trials or not, and in many times in the gene therapy world, we don't require randomized trials. We need a fit-for-purpose single-arm study that's based on either very good natural history or very large effect sizes. When you have large effect sizes that you can see a big difference in either a biomarker, as was just mentioned, when you have a biomarker that you can measure, or in a clinical effect, such as when we saw, for instance, with SMA1, Zolgensma, where in 6 months you knew from the clinical outcomes in 6-month-olds that were treated with that gene therapy that you had made a very large difference. You don't need a randomized trial, you just need an observational trial. So there, although there was reasonable natural history, one didn't actually even need it because one could see over the course of the clinical trial that you were very far off from what the natural history of progression would be. So I think designing trials end up needing to be very much fit for purpose for the given disease that you're dealing with. And some of that has to do with what we're talking about already. Do you have a biomarker that you can hang your hat on? Do you have clinical outcomes that you can understand well enough to distinguish as being different without needing a randomized control arm? The randomized controlled trials in this area are often very challenging because of the small populations. On the other hand, some people use of trying to use historical controls can also be very challenging because the standard of care, even for rare diseases, sometimes has changed over time. And so we have to be very thoughtful when we design these trials. I think, you know, I would, before I turn it over for to kind of to a variety of questions, I'd just say that this is just one of these areas where when you think about it, you know, part of the challenge is actually taking the time to think about what will make the most sense. It's very easy sometimes for companies and others to run very rapidly into wanting to move ahead and start a trial, or for that matter, foundations, and so not sufficient thought is given to the end game of what data will be needed to get towards a regulatory approval. And sometimes that it helps to slow down a little bit and work with regulators to make sure that even from early phase trials, that there will be careful thought given to what's going to actually get over the finish line, particularly in gene therapy, where sometimes one trial is all that's necessary, because if the effect's large enough and the study is well enough conducted, you can actually have one trial that could end up bringing a product towards registration. So I guess, I'd be interested in others' thoughts. I think we clearly will only do double-blinded, placebo-controlled trials now when it's kind of we've kind of come full circle. Originally, they were held up as the sine qua non of what was the right thing to do. Now, actually, we use them as the last resort when you simply, we only will do those when there's no other way we can get there using a biomarker or a large effect size or some clinical endpoint that we can distinguish. But I'd be interested to hear of others. I mean, the problem is we still sometimes end up in this realm of needing to do these. The one that immediately comes to mind is not a pediatric condition, but just as an adult example, amyotrophic lateral sclerosis that's not hereditary, the 90% that is acquired, or that appears to be acquired. We still have so little understanding of the underlying pathogenesis. The outcomes are so variable, and the therapies that have been able to be implemented to date are less than overwhelming in terms of their efficacy. It's an example of where we've had to fall back onto double-blinded, placebo-controlled trials still. That doesn't mean that they don't have best supportive care with other drugs at the same time, but that's been an issue. So happy to throw it over to other panelists for comments on this. I mean, I think at FDA at least we've come to a place of accepting in the gene therapy world that whenever possible we don't need randomized controlled trials as long as we can feel convinced that we're truly seeing an effect size.
Kristina, do you want to add anything? Or is she muted?
Unmute myself. Sorry. Very little. I mean, I think from our side, it is very much the same. We also accept single-arm studies, and it is indeed the effect size. If it is huge, of course we accept it. I mean, everybody accepts it then. Unfortunately, in many situations, maybe not so we've seen in gene therapies, but maybe in other type of products, it is not always that the effect size is that big. And I think maybe some people know in Europe we like the active controlled studies if possible. And actually we have seen some rather recently in Pompe disease where one new enzyme replacement therapy was actually tested against another. So a very fortunate patient population where there are several enzyme therapies. Of course, like in Sanfilippo, there is nothing so far, so you wouldn't be able to do such a study. But yeah, so from a regulatory point of view, when the effect size is big, there is no problem from our side in Europe either to accept single-arm studies. Although control is, if there is some sort of control, it's always useful, as my statisticians used to say.
The statisticians.
I mean, so as well as developing our own therapies, I've worked in many early and late stage clinical trials from pharmaceutical companies, and whilst I accept there has been a significant movement from the regulators towards accepting more novel trial designs, I mean, I'm involved in two ongoing trials where there is randomization to a current therapy that we either know is completely ineffective for one element of that disease, for example, the CNS, or significantly more toxic than the currently than the IMP that's being tested. And so I think I accept that there has been a lot of movement from the regulators, especially with the accepting of accelerated approvals. I think we're still being asked to do, as clinicians and families, to do trials that none of us have any equipoise over in the community. That I cannot sit in front of parents and consent them to studies where I believe that either therapy would be delivering, some benefit to the patient, and that I don't know as a clinician that neither is better than another, and I should have a balance and equipoise. But in several of ongoing trials that we're recruiting to, I don't have that at all, and I know that one therapy is either entirely ineffective for that aspect of the disease, or another, the comparator, is actually more toxic, and that's very challenging. Our patient communities are very educated, aware. They come to me for consent, knowing this, and I'm honest, and so I think there's still quite a way to go on this, and my concern is someone who does a lot of pharma studies, but I'm not in pharma, is the conversations that happen about the trial designs happen between pharma and yourselves as regulators. They don't involve the wider community of clinicians and patients. If we're given in a rare disease a trial as the only way, this is what's been approved, this is the only trial that we can do, then yes, as clinicians, I'll try and recruit to that. As patients will accept a lot of risk, but that doesn't mean that's the best trial design, and it doesn't mean that we're happy to be accepting that risk or that there isn't another way. And sometimes it feels like we need to somehow be able to have these conversations about what is the best trial design that involves the wider community, rather than, perhaps a new biotech who's been in the field for 3 years and comes to you guys, when the patient group may have been working on this for 20 years in the background. We need the wider community involved in these discussions, because we often only get one chance at doing these studies.
I totally agree. I would like to invite Trista to talk about this, early participation of patient groups in clinical trials.
I'd like to say to everyone that's in the room that is doing any drug development in rare disease, thank you. Thank you from the patient community. I remember a day 20 years ago when no one was doing anything for our community, and it was a dark time. I had a daughter that was 4 years old and didn't have the outcomes that we know families will have today, so thank you for that. The one thing that I can say is if you are developing a drug, get the patient advocacy group at the table in the very, very beginning. Like day one, get us to the table, let us explain what's important to our families, let us also tell you what will work and what will not work. It will save time and money. Also, help us educate our patients. If we can educate families along the way, when it comes time to recruit, there's not this big curve, because they've been a part of it, we've been a part of it, and they trust the patient advocacy group, which allows patients to feel more comfortable. That's number one. Number two is patients come to trials for different reasons, and understanding those reasons. So for me, my daughter has OTC, and there are lots of options for OTC today. So we have to find which one fits for our family. My daughter's been transplanted, so these options aren't for her, but for all the other families, we have to find what fits. Now, for some of our other conditions, there are no options. So what is available is what they'll choose. So you have to understand some markets are crowded and some are not. And I love that they're crowded. That's a wonderful problem to have, but it makes it to where families can choose, which is choices are great. When it comes to the design, we have to make sure that families It's difficult enough to live with a rare disease, but to also put a trial on top of it, even though it's a benefit to the family, it can't be the burden of the family. We did the Ravicti trial 11 years ago, and it was a burden, but it was a burden we were willing to take. Some families, I think it's generational too, some families are willing to, if you have an option, they're like, we're in. Other families like to sit back and wait until something tragic happens, and then they go, okay, we're in. So you have to understand there's different reasons. You want to go? Okay.
Thank you. Teresa, you want to add to that?
Yeah, I just want to add that, as Teresa said, it's very important to evolve since day one. Maybe not in gene therapy like Dr. Mark said, but other therapies, the regulators require a randomized controlled placebo trial. And for example, as a mom and an educated patient advocate, I will be very, very reluctant to put my child on a trial like that, because in these diseases, they are degenerative. They don't stop. They lose abilities every day that passes. They get worse. So, and there's a learning window, and there's a time for that. In putting a child into a placebo, it means that there's not ethical from my point of view, because they are still losing abilities anyway. So also the issues about the placebo trials is that some methods of delivery are very invasive, and sometimes you need to go under surgery to put the devices on, to get the drugs, and I cannot see how can we convince or get patients into a placebo trial where there's such complications and risky procedures like this.
Yeah, I would like to echo on that point and go back to the regulator. We hear clearly that a double-blind, placebo, randomized control is not necessary for gene therapy, and we're very happy to hear that. But there are many therapies for somewhere in between, and there's the ethical dilemma that was being brought up by the patient advocacy group here, particularly concerning pediatric patients. How do you convince a parent to let their child to go into a double-blind, randomized placebo trial? So I want to hear if there's any other comment on that, particularly from maybe the FDA and the EMA perspective.
Yeah, so this is obviously always a challenge, and some of it's understanding, again, the effect size that you're seeing or expect to see with your drug and the ability to determine outcomes. Ideally, you have a biomarker or something that you can get a handle on that will make it so that you don't need a double-blind, randomized, placebo-controlled trial. In some cases, one doesn't have that, and you end up there because it just turns out that a randomized, double-blind, placebo-controlled trial is the most efficient manner of actually getting an answer. But I think we do that as a last resort when everything else has not worked out, and I think that's what you have to explain. I mean, you have to try. We have to try everything else we can, use every other tool in the toolbox, and once we've run out of those, I think then you need to be able to explain to a parent why we've run out of all of those other tools, and we're left with a situation where you're going to be having a situation where best supportive care plus or minus some intervention is what you can do. And I do believe it is something we try to do as a last resort. But it is true that sometimes it becomes necessary, usually when it's for for products where you don't have super clear efficacy or a good understanding of the underlying mechanism of action. But maybe I'll turn it over to my European colleagues.
Yeah, no, I agree. I absolutely agree. We do not want to do it if it's not really needed, but almost all, I think, pharmaceutical products have also side effects. So if it turns out that a product is actually not efficacious at all, but causes side effects that you don't want your child or you don't want the adult person to have, then in those situations, I also see the reason to actually do the placebo-controlled study, because you don't want to license a product that possibly may do something on a biomarker but turns out to be very unsafe for the patient. And that's obviously also some of the benefit risk assessment is very much part of, how safe is the product? Can we give it to the patient? So it can be super, super efficacious, then it can also be possibly rather unsafe, but yeah, it's part of the package we need to know, both the safety profile and the efficacy profile.
Yeah, thank you for those comments. I mean, I know that the regulators are coming from the perspective of protecting patients, and we really appreciate that. But I was wondering whether it has to do with some sort of historical context. Our regulatory approval process started with common disease, and these affect adults. So randomized placebo-controlled trial is the gold standard, and there's no question about that. But when you go to rare disease, they affect children, and that the ethical question of control become much, much more sticky. And so we're just trying to sort of bring that conversation here, and then we can all think about, good solutions and good compromise going forward. The patients are safe, and trial will not fail. I would like to, if I may open the floor, because I know some people on the phone want to speak. Yes, Marshall, please.
So first off, thanks, Dwight. First off, love the comments from everyone. It's been a very, very good session. I think the point you just made, Barbara, is a very important one. There's sort of a legacy issue around, patient safety comes first before all else, efficacy and everything else. And there's a real disconnect between that and what we see in the rare disease population around our tolerance for risk, patients' tolerance for risk. One of the ways to get around that I don't see people taking advantage of, and Teresa made the great point that involve the patient organization as early as possible, but also involve the regulatory agencies as early as possible. If you come to them and throw a protocol on the table, they may have to revert to kind of the base level, which is, well, safety first, everything else after that. If you start early, and both agencies have great mechanisms for having non-binding, informal discussions where you can talk about the science, you can talk about the design, and actually get really good input that I think later on can lead to a much better clinical trial design. Nobody at the agency says, I don't want a child with a lethal disease to get treated. They just don't. But at the same time, if you haven't educated them, had the dialogue with the patients present, with the investigators, with the sponsors, and had a very open dialogue, you tend to get these much more rigid designs. And that's been my observation. There's an interesting statistic. There were two really good papers that looked at why do clinical trials fail. And they looked at over 3,000 of them. And there's two separate studies, one from the biodatabase, one from clinicaltrials.gov. Interesting, only about 20% of them fail because of the science. Science is usually pretty good, particularly in rare disease. Most of it, it's around recruitment and retention, which is study design, having realistic expectations for the population, having worked with the patients early. So it really is more of a team effort than any other field I can know of.
Thank you very much, Marshall.
Just follow Marshall's comment with a related one. So we have the good problem in many diseases of having an available therapy, and then the dilemma is coming in with a study drug, and how do you do a trial to show benefit? So if you have to enroll patients never treated, that's a diminishingly small population. But if you have to stop the therapy, wait for the patient to decline, then give study drug, that may be questionable. Or if you stop the therapy once on the study drug, once gene therapy is done, and look for the absence of a decline, you might not get there and you might not retain the patients. Does that mean that we have to do a double-blind placebo-controlled trial? However, that also has the dilemma of, is it ethical to, take patients presumably off of the study drug in the comparative group? I'm not sure how to solve that one.
Hi, Alessandro Prigione from Düsseldorf, Germany. I wonder if you can comment instead, what about crossover trials, maybe with multiple cross, and also about N=1 trials, where every patient has his own control, so that especially for those very heterogeneous diseases where the discourse is very different, one can follow up a specific patient and then compare, to its own. Yeah, thank you.
Addressing that to either Peter or Kristina, I assume? Is that what you're asking?
I can take a stab at starting. I mean, the N=1 trial brings up this issue of if you have, and this is why I think the comment about coming into regulators early and often is a good one, because in some cases, we can suggest to people that they start gathering run-in data for clinical trials for the 6 months while they're discussing the trial design with us, because if they actually get baseline data, one can then look for changes from baseline and not need a randomized trial. So in some of that's taken to its natural limit in the N=1 case, where you're trying to look at some baseline functioning and then show that after an intervention you've seen some change in outcome. Now, obviously, there's not a lot of power there, but it's for the N=1 approach, I think it becomes whether you believe in the principle of the design of the therapy is working, whether it's antisense or an N=1 gene therapy. And as long as you have confidence in that, one can potentially proceed. I don't know. Kristina, any comments on your side?
So the N=1, I have to admit, I still haven't seen any discussions on that at the agency. We have had no scientific advisors or protocol assistants or discussions early on with any applicant developer for an N=1. We have, it's been discussed in meetings, etc., but we still haven't seen it. So it's difficult from a regulatory perspective then to say this is actually what we would do. Yeah. So I don't know if you have at the FDA seen
so we've had there are some people in the U.S. who have done this for again, it's usually on the antisense oligonucleotides for N=1s, and we've had more discussions around the process, because it's really what you need to do to show that the product is safe enough to give to an individual, and then how you might try to assess for what ends up being really an anecdotal outcome and hopefully the idea is if you put together then enough of these N=1s, you'll show that the principle, the approach of design something that addresses a molecular defect leads to an improved outcome, is that principle works.
And crossovers?
He asked about crossover designs. Your thoughts on those?
All I can say is that that at least I'm not the best person to talk about them, because in the gene therapy world,
doesn't work.
What that ends up being is just delayed administration of gene therapy, you basically, it means that you have a run-in period where one arm gets treated and then the other arm, because you can't take away what one giveth in terms of gene therapies. It is something you could do with an antisense therapy with a washout period. Although there, I'm not aware. Again, I'm not as familiar with all those products, but I'm not aware. It's not a design that we've seen a lot of in our center.
No, neither have we. I mean, we see both the chemicals and biologics, but No, multi-crossovers, I think 1 or 2. I guess it also becomes quite complicated from a statistical point of view, and to sort of calculate what in the end actually was the effect. But having said that, I mean, if you really would consider something that has a lot of crossovers, then indeed early discussion with the agencies you think would be warranted to try and, in that case, just exactly how long would the washout period be, or between the crossovers, to ensure that you could actually capture something. So yeah, I would, yeah, again, I'm not a statistician, so I would also say that please do, if you plan to do such a study, come early and discuss it.
In light of time, we will move on to the last 2 topics, and we'll come back to the Q&A and see how much time we can stretch. So the next topic is how do we increase patient participation and recruitment in clinical trial? We already touched upon the importance of early involvement. So maybe if I may ask Teresa and Raquel to share, what are some of the practical points that you've seen in trials, then you say, aha, that doesn't work, or like, oh, that really work, and then for the audience to hear.
Okay. For urea cycle disorders, clinical trials have to fit, number one, into what the family is looking for. When it comes to the actual recruitment, recruitment is going to be important to come through trusted sources. Make sure, I know we have some instances where recruitment is coming from social media and not necessarily through patient advocacy or clinicians, and sometimes that poses a problem. Families will often ask, why hasn't our physician told us about this clinical trial, which is a red flag for families. So again, making sure that you come through the patient advocacy group, but also the clinicians, the trusted clinicians that are part of the, maybe the medical advisory board for the patient advocacy group. And then also making sure that the trial fits into lessening the burden for families, making sure that families can participate in a way that doesn't disrupt the family. I know that many of our families, it's a financial burden just to have the condition. And sometimes trial designs will say, well, we'll pay for travel and for lodging, and that's wonderful. But sometimes you have parents that are not able to leave work because they have a position where they can't have that time off, or there's other children in the family, or just the whole family dynamic. Being able to do maybe study visits at home, that would be wonderful. I know it's expensive, but that would be another really great way. And then I think I can say for OTC, we have a lot of options, making sure that we can educate families on which option works best for them, because one size doesn't fit all. And families have to make the decision now between gene therapy, stay in medical management, and liver transplant. And so being able to assist families in making the decisions to participate in trials or waiting maybe for the next one or for one that's in the pipeline, many families are kind of struggling with that right now.
Regarding patient involvement, I think it's very important to be transparent since the beginning about what does it mean to be on a trial. There are many challenges in patient recruitment. In these diseases, the companies tend to choose and prefer asymptomatic patients, which is tricky because diagnosis happens often in a symptomatic phase. So unless you have siblings of symptomatic patients, and because we have no newborn screening, this reduces even more the patient population for recruitment. And really with the also exclusion criteria, is usually, it's having taken, if you have taken part in previous trials, it's also, they are also excluded. And as I said before, with so many trials failed in the last years, it's even harder convincing families to participate. It's very discouraging because we don't have examples of success that we can give to families. The examples we have is families that made huge sacrifices, moving home, traveling far to usually one trial spot, large travels, the burden on the siblings, and then at the end the trials end up closed, and the families are left abandoned, kind of abandoned when the trial ends. So that is discouraging to, convince families to participate. Also, as I said, the neurodegeneration happens very slowly. And the variability of symptoms is big from child to child. And so designing the trial is really difficult, as I said, and because it turns difficult to measure the changes during the length of the trial. So what we think is the traditional approach, it's poorly suited to degenerative diseases. And these neuropathic disorders should have different approaches in trials. So that's why we ask for regulatory flexibility. So what I want to ask for regulators, and my message is for these neuropathic disorders like Sanfilippo. These diseases are very difficult, are very complex. It's a form of childhood dementia. The children born apparently normal, then they lose their abilities to eat, to walk. They suffer a lot, pain. They experience seizures. They have abnormal behaviors, and these diseases that are very hard to manage from a family perspective. They are also fatal, and we have high unmet needs. So that's why we need different approaches like emergency use authorization or accelerated approval, because CSF, heparan sulfate, is reasonably likely to help children. That's why we think. So we hope on the regulators to listen to us and weigh all the risk and benefits, because I still hold on hope, and I don't want to lose hope.
Well, thank you very much for this emotional sharing. I'm sorry, we'll pause for question for now until we get to the end, because we're running a little bit short of time. But I do want to really thank Teresa and Raquel for your sharing. It's very valuable, and I think particularly for everyone here who worked so hard to develop this disease. And one of the beauty of this discussion is that I know scientists and clinicians work really hard. I know the sponsor worked really hard, and I know that families are suffering, and we're trying to see how we can bring everybody here, everybody's perspective. So we'll move to the next one, which is hope, as you said. We want to hear what is the advancement in regulatory framework and what's the possibility of global collaboration to move the field forward. So, Peter, would you like to lead that?
Yeah, no, thanks very much. So I think, the COVID-19 pandemic made clear that there are many things we could do better as regulators in not just in infectious disease areas, but in the rare disease space. And I think two key areas that have become clear, especially in rare diseases, is kind of an urgency towards regulatory convergence, and then the potential benefits of increased agency sponsor interactions. I'll start with the first of these, which is that it's very clear that with these small populations, the barriers that have been set up, in some cases just because we haven't taken the time to harmonize certain things like gene therapy requirements between the US and the EU, have led to barriers to entry that are preventing, for instance, a gene therapy that's available in the US from becoming available in Europe or vice versa. And so with that in mind, we have engaged in a dialogue with EMA to try to resolve some of those. We're trying to engage in this more global discussion. We've taken this up also working with WHO, because ultimately we shouldn't just be thinking about high-income countries. We also need to think about low and middle-income countries and how we will get these therapies, gene therapies and others, into those areas as well to bring benefit globally. So I think it's been a very good conversation, and a little bit shockingly or surprisingly to me, in some cases, when we've actually put people in the room together, we've learned that the reason why we had differences, which were perceived as irreconcilable in some cases, it turned out to be simply that, well, we just did them differently to start, and nobody actually bothered to get into the same room and discuss them. That may not be true for everything, but I do think that this is a very worthwhile discussion, because this concept of rework, it's actually it's against the public health needs of any given country, right? Because if the disincentive to help rare disease patients because of perceived regulatory barriers of entry is not a good thing. So we will continue to work with our colleagues. I can't promise it's going to be the fastest going always, but we're trying to do it in a manner that is as speedy as we can. The second piece I will say is something that is, I think, our European colleagues, we have realized, I think we've had these conversations, that what's come up multiple times today is the importance of sponsors coming in and talking with regulatory agencies. Over the past few years, one of the things I take pride and joy in is that by encouraging these early conversations, we've helped sponsors avoid doing unnecessary toxicology studies. That's good for multiple reasons. It saves time, it saves wasted killing of animals, and it moves the field forward. Plus, this new kind of concept that we can have more ongoing dialogue. We have a pilot program that's ongoing at FDA, both in the Center for Drugs and the Center for Biologics, where in the Center for Biologics, we have 4 programs where we're having, instead of the normal formal meetings, which usually take 30 to 75 days to schedule, we're having an ongoing dialogue with the product developers such that they can contact us at any time with questions, with the concept being that this kind of ongoing dialogue may allow us to significantly shorten the time to development and avoid more missteps. So hopefully the combination of enabling development and unifying regulatory requirements will help move these products through regulators more quickly. I'll turn it over to Kristina if she wants to add.
Yeah, I just wanted to say as well that, of course, because the EMA and the FDA, we speak a lot with each other. We have what's called cluster-specific sessions, one on rare diseases, for example, where we can talk about advices given, marketing authorization procedures, products in development, etc. So it's a super good forum to try and harmonize our views. But what sometimes happens is that indeed a company would come to the European Medicines Agency, they would say, this is what we're planning to do, and we say, oh, okay, looks rather okay. And we accept it. It's not like we're going to tell them you do something completely different, and then they go to the FDA and say, this is what we're going to do, but haven't sort of harmonized in advance. And if the FDA then says, no, we need another endpoint or a different duration of time, we do have the possibility to talk to each other as well. But there was one thing I was thinking as well with regards to patients, and because of rare diseases, there's so few patients across the world, of course, and there are certain regions in the world where patients from that particular regions are required for regulatory approval, which obviously can make it a bit more difficult if you don't have patients in that region. If you haven't done the study there, maybe you've done a study in other parts of the world, but it's still a rare disease. And I think here it obviously would be useful with the patient input as well, to say that, are you willing to accept data? So I'm from Sweden, I'm willing to accept data from France. I don't expect patients from Sweden to have been tested because maybe there are none. I'm fine with the French patients. Now, of course, that's in the European Union, so we have to accept it. But still, the concept of it, I think, is very important. If physicians and patients as well are willing to accept data from other regions, because that also may help a little bit both the developers and indeed progressing faster. That's more, a bit of a question also to the patients and physicians, maybe. Thank you.
Really quickly then, a word for data sharing. Preclinical data sharing will prevent duplicative pharm tox studies. Clinical data sharing, many of these agents are related, and so a problem for one may have implications for many of them.
Well, we just wanted to thank everybody for participating. We have a lot of work to do and lots of problems to solve, but hopefully with continued working together, both pharma and academicians and patient groups, which is obviously very key, and the regulators working together, we can keep making forward strides. And thanks everyone for participating.
Thank you very much.
Thank you. Bye.
I'd like to particularly thank Peter and Kristina. I mean, I think it's a very good session, and we have a good discussion, and we hope the community will continue to thrive. Thank you very much. Thank you.
Bye.